to the Weekly Bioanalysis, a KCAS podcast.
Hello and welcome to the 13th episode of the Weekly Bioanalysis. My name is Dominic Moreno and I'm the Senior Scientific Advisor for all things related to ligand binding assays, biomarkers, and flow cytometry. I've been with KCAS for about 7 years, but I've been in the bioanalytical space for close to 25 years. I'm here with my co-host, John Perkins.
Hi, I'm John Perkins, Senior Scientific Advisor for all things related to LCMS. I've been with KCS 9 months now, and I've also been in the bioanalytical space around 25 years.
So today's podcast, as always, is brought to you by KCAS, Kansas City Analytical Services, servicing the pharmaceutical and biotech industries for over 40 years. John and I are senior scientific advisors, as we've previously mentioned numerous times now. Either or both of us are available to answer any questions you may have regarding this podcast or any of KCS services. We are thrilled to have you listening again as we get ready to celebrate another episode, this one being our 13th episode. For those of you that are interested, you can find all of our episodes at kcsbio.com. Also, as a reminder, you can follow us on Twitter at The Weekly Bio and at KCS Bio. As we mentioned before, we started this whole thing because of social distancing and getting away from our normal means of communication, which was, getting out and visiting people. Even today, John and I are in completely different places. I'm in Kansas City. John is in upstate New York. And our producers are in different places around the Midwest.
Except we're not producers this week. We're down to one producer because, and this is why we took a time out last week, was that our second producer, Erin, gave birth to a son last week, and she will not be part of this production for the next few months. So congratulations to Erin and her husband, and we look forward to welcoming her back in October.
Yeah, mother for a second time. Congratulations, Erin. We hope you're listening. Similarly, as for why we missed last week's podcast was Jeremy, our other producer, had been in Florida having a nice vacation. So we're glad to be back. So as we mentioned, this weekly podcast will give us a chance to get to know all of you. And we hope it gives you a chance to get to know us. And even more importantly, it gives you a chance to get to know KCS and the services we provide. As always, the podcast will be a review of the latest news and resources, and then a focus on a topic of our choosing before discussing any feedback we've had. We're constantly looking for topics, and we'd be happy to discuss something that you might want us to cover. So go out there and do some posting, and we'd like to hear from you. And then we will wrap up and give you a little teaser for next week, letting you know what is coming up in the future. So again, we're thrilled to be here. This is our 13th episode. It is a jam-packed episode. We are so excited to have everybody. It's going to be a fun episode. We've got a couple guests, very important guests, very knowledgeable guests, both Lawrence Goodwin, our Vice President of Operations, as well as John Pirro, our Senior Director of Ligand Binding Assays. We are so excited to have them here to discuss part 3 of our series of 4, and that's the overview of PK modeling and the impact on bioanalysis. To kick us off, John is going to go over today's podcast topics before we jump into the news and resources. John?
So yes, we're going to follow the same structure as usual. Next up will be the news items with the focus on COVID updates, and then non-COVID related items too. We'll then move into the main portion of the agenda, which is, as Dom said, is where does bioanalytical, part 3 of 4, and where does bioanalytical fit in the drug development process? This time we're looking at overview of PK modeling and its impact on bioanalysis. Then we'll discuss some feedback from you, our listeners, before teasing what we're going to be discussing next week. And so let's move to the news. First up, it seems there's a lot of activity on the vaccines front. So we have Moderna have reported positive phase 1 data, and then the AstraZeneca Oxford University vaccine should actually have data in the Lancet on Monday. I know. Looking at articles about it, the Moderna vaccine is definitely showing that they're seeing antibodies to the vaccine, whereas Oxford are talking they're seeing both antibodies and T cells with hopefully optimism for the longer term nature of that vaccine versus something that's showing antibodies only. Any comments on that, Dom?
Yeah, certainly those two are emerging, aren't they? We've talked about this quite extensively throughout our podcast history. It's interesting to see that these are sort of the front runners, although we shouldn't discredit the Regenerons and the Pfizer BioNTech and the Boehringer Ingelheim and a few others in there, Merck most notably, and Novavax and even Lilly. But like you mentioned, the AstraZeneca Oxford one, that one's really, it's a dual antibody. It's against a couple different proteins, COVID proteins. It's certainly got the lead. They're showing some real nice efficacy, and like you mentioned, there's a publication coming out here soon that would be interesting. And then Moderna, Moderna is an interesting one because they're constantly, it's their stock seems to always be impacted by it, right, John? So they have these announcements and the stock goes flying up, but it is an mRNA based one, and they seem to be right neck and neck with, at least they demonstrated some clear neutralizing antibodies. So all very positive. But then, you sent me one other article that was the CEO from Merck just saying, hey, this could be a huge disservice. And as you know, I've always been somewhat skeptical about having, vaccines ready in 6 months, right? So, despite all this, look, I'm cautiously optimistic. We've never had, this is a historic run at vaccines, but I still think there's quite a bit of research that needs to be done. What seems to be happening is we're probably going to jump a little early and get everybody, I think they're talking about vaccinating upwards of, I think it was 30,000 people with the first salvo added after it gets through the next phase. So I think it's 3,000 and then up to 30,000. So, I guess it's, overall positive, John, but, I still think I think we need to temper that expectation about having an effective vaccine before the end of the year.
Yeah, I think there's so much attention to it is that it's really easy to get carried away on how quickly things are moving. I think the interview with Ken Frazier was really interesting in terms of trying to add a dose of reality, particularly saying that Merck have, I think they've introduced four of the seven most recently introduced approved vaccines, and the quickest that anyone's ever done is 4 years, which was Merck for I think a mumps vaccine. So yeah, I think there's obviously so much attention as people wanted to move quickly, but I tend to side on the let's be a little bit more cautious about how soon we'll see something to really address, have active vaccine available for people. I mean, I
Yeah.
think Moderna is interesting. We talk about their share price. They're always in the news in terms of data, and then it becomes their manufacturing issues, which they now seem to be slowly resolving. And I think there's some of that is the relative inexperience of Moderna, who've never had an approved product, versus mature companies like AstraZeneca and Merck and Pfizer being in the vaccine development, and the different approaches resulting from experience of doing it.
Yeah, that does make Moderna sort of I don't know if dark horse is the right word, but having never had an approved one, there's a couple other things before we move off of vaccines. One is the efficacy, right? So there's talks of it. Does it need to be 70%, and then it's lowered down to 50? So I feel like the rushing of it that's part of that disservice. We're lowering the expectation of how good these vaccines need to be, although I guess if it's presumably a safe vaccine, which that is something we can debate at another time, then I would have no issue with giving it to myself or my family, knowing that it might only be 50% effective.
And then you obviously have the longevity, which we can't assess this quickly. I mean, there's been reports of antibodies die off quickly. You mentioned T cells are another part of the immunity piece. So this is where the Oxford thing think they show benefits. So yeah, we've got no long term data on this at all. So yeah, who knows if you could have a vaccine that's effective, but if it's antibody based and dies off quickly, it might not be of a long term benefit to have that vaccine taken.
Well, that was one of the real powerful things that I read about Oxford's and AstraZeneca's vaccine is it does seem to produce some nice, long-lasting immunity. And then a great read on the CEO of Oxford. I believe she was the CEO. But that was a wonderful read you sent along. But John, we're gonna move on to treatment updates. Maybe let's jump right in. I thought this was fascinating to read that remdesivir, which got all the praise, right? That's the Gilead drug. I think it's still the only approved drug. That's only given to 1 to 2% of all patients. I thought that was really, I don't
True.
know. And then dexamethasone was 2%. hydroxychloroquine, our friend, is still at 1%. And then the number one treatment is still an antibiotic, erythromycin, which is at 28%. That doesn't make any sense. That's not treating the virus. So I feel like there's just so much misinformation out there, and even clinicians seem to be behind, right?
I think you're right. I mean, I think dexamethasone. I'm not surprised that's only 2% because at least what we've seen it's only supposed to be effective in late stage treatment, so that will be a much smaller proportion of patients. The azithromycin is just incredible. I mean, we haven't seen anything to back up any use for that at all. So for that to be the most prescribed drug is just bizarre.
It's like they're living in another world, right? Some of these physicians are just they're like, "Oh, I'm just going to keep throwing that right hook that I've always got, and I'm going to give them erythromycin, and that has absolutely no benefit for a virus." So
Yeah,
that
yeah.
was, I don't know, that made me scratch my head quite a bit. So, the last item on COVID was, yeah, this was an interesting read, right? So the FDA complicates chaotic federal COVID-19 response with pledge to resume domestic inspections. So I do like the idea of the FDA starting to inspect, to get back into the norm. But during the COVID pandemic, it seems kind of an awkward time for them to, especially when we're seeing such a spike in cases, to have them come out and say we're going to start visiting facilities. I don't know really what to make of it, but I guess it's a good thing. I don't know.
I think is well, I would think it's beneficial, but yeah, they're going to have to really define how they're going to do it. I mean, but I know with us seeing remote audits, to me that's really the way to be doing it for right now. And yeah, the FDA obviously have to figure out how they do it. There's got to be a lot of practical implications to try and get out and actually visit facilities.
Yeah, that was part of the read, right? They said they felt like they couldn't quite get a good handle on what was happening virtually, so they wanted to start visiting sites again. So yeah, I mean, it's if we hadn't seen these massive spikes, and we don't need to get into some of the states that are still very problematic, and but this would, to me, would have been a real positive sign, right? I like the
Yeah,
idea of starting to get out there again, but John, on a side note, we just found out here in the state of Kansas that all schools have been pushed off. We're supposed to start school August 17th, and now that's pushed back to September 9th after Labor Day, and they've suspended all sports temporarily. So, I had mentioned my son had started conditioning in football, and that's been suspended, and they're trying to figure it out. And mind you, this is outside on a track with masks on and 6 feet away from each other, and they even suspended that now locally. So some odd times.
yeah, it really is. I am very much in favour of the careful approach and mirroring how we've done things at KCS. I really think that's the right way to go. I mean, I heard last night there's a few more cases in my area, which we've barely seen any. So I'm waiting to hear more about that. But I don't know anything about schools reopening, etc. That hasn't impacted us here in terms of my family. So I'm not sure what the plan is right now for New York.
Yeah, this is rocketing. This is late breaking. It happened literally late yesterday that the state of Kansas has issued a moratorium on sports, and starting school basically a month later, which I don't know. I'm fine with that. I always thought it was weird we started so many weeks ahead of Labor Day. So there's really, I don't know what the impact is, but certainly, we're taking a cautious approach here in Kansas, and that's a good thing. So
Sure. Should we move
um,
on to the non-COVID?
yeah, you bet. Why don't you want to start us off?
So you sent the link, and I've only just found it. At least going to the FDA site, that they've added 5 more products to a list of hand sanitizers, of which there are 59 which are being recalled for containing methanol. I thought it was interesting just going to the FDA site, and they looking at the FDA warning consumers of risk of methanol contamination in certain hand sanitizers. And I love that if you read down, the agency is especially concerned of danger of drinking any hand sanitizer under any conditions. I'm not sure what would drive anyone to do that. And obviously, having methanol in it is much is a major problem. But yeah, it's not high on my list of priorities, I have to say.
Well, John, there is like a 50% spike in alcohol sales, so maybe there's a lack of stuff on the shelf, and
But,
people
but
want to drink the hand sanitizer. I don't know.
considering how hard it is to find hand sanitizer, I mean, we barely see it here, and certainly what we do see is bottles that I don't know who the manufacturer is. It's almost like it's popped up out of nowhere. So I can see where some of these risky hand sanitizers are coming from.
Oh, they're everywhere. There's actually quite a few of them here, and some of them have some real bad smell. I mean, there was one. I'm convinced I've touched one that was definitely contaminated, and like immediately was like, "Don't anybody use this," right? So there's some of them that when you and they're very nondescript bottles, and they tend to be these big gallon jug pumps, right? And
Yep.
it's really loose, and it's like again, it has a very tainted smell to it, and that. I mean, 59, man,
Yeah.
that's a big number.
Yeah, I was in the supermarket last weekend, and they had a thing of hand sanitizer as you exit, and it was a brand name which I really like the smell of. I mean, it's great. Get that whiff all going off in your car, and so I put it on my hands, and you're right. It was. This is not what it should be. It was horrible. So it's like now I've got my hands that I really don't want to be touching anything with. Anyways,
Yeah.
let's move on to this is, I think, something that you found.
Yeah, this was, I love my cell therapies, John. We can't go a podcast without that. This was a really neat thing. So it's a safety switch based on nutrient deprivation. So, as we know, there's some side effects to some gene therapies. They frequently might cause some adverse effects that manifest themselves in a cytokine storm. Although they are incredibly effective, they can have this somewhat, well, not somewhat, definitely a big adverse effect of them. And now they're finding ways to do some way of depleting nutrients from them, which is more likely going to turn them off. So really cool engineering and always something worth noting.
So this is early days, though, because this is what had been done in mice. So still got a fair way to go in terms of investigation. But yeah, really, really interesting stuff.
Progress, John, right?
Yeah,
This is a really great. I think it's a wonderful way. And I sometimes find myself thinking about things like this and how are they going to overcome this? And this seems like a real viable solution to be able to have a way that the engineered CAR T cell requires a higher nutrient level, and you can kind of turn it on and off. So
nice.
more to follow, like you said, but really, really cool stuff.
Yep, absolutely one to watch. And finally, we talk about this is it's a great story. This one it's actually in London. There's a care home where the residents have been under lockdown for 4 months, and the person in charge of really trying to help keep the residents entertained has had sessions where the residents are doing mock-up of album covers. So the some of the instances are the Aladdin Sane, David Bowie Aladdin Sane cover. We've got Adele 21, Bruce Springsteen Born in the USA, and they've actually got the residents to pose in very similar poses to the original album covers to show them side by side. Queen 2 is a great one as well. I just thought this was a fantastic thing that the guy was doing, and he's really got the residents of the home all sort of fired up about it too.
Yeah, it was great. I had such a good laugh looking at that. Blink 182 was one of them with the nurse with the needle. I mean, it's so like I love these types of stories because most of what you're hearing is people are stuck in nursing homes. There's a lot of it. We've talked a little bit about some of the effects that this might be having, not only on like lack of circulation because they're not moving around, but these sort of mental or psychological effects that are taking place because you can't go visit or grandpa for things. And now you hear this story where someone got really, really clever. And I hope everybody, it's called the nerdist.com article, right? So if you can go and check that out, it's
Yeah,
pretty cool to see. It was like Adele 21 and it had like Mabel 93 next to it, right? It was real. I don't know if it was, but it
yeah.
was really funny to see it that way. It was great.
Yeah, massive shout out to the Sidmar Lodge Care Home in London, and the guy who
Yeah.
actually coordinated it is a guy called Robert Speaker. Yeah, it really having had relatives in care homes, that you're right, the mental stimulation piece is huge. So to find sort of fresh activities for people to do is just excellent. Really, really impressed with that.
Yeah, that it was it brightened my day to even read about it. So Well, John, I think we're wrapping up our news and resources, and what an exciting 13th episode we have today. We've got a couple guest speakers to walk us through some really great stuff in terms of PK modeling and the impact on bioanalysis. So, John, do we want to jump right into the main portion?
Yes, let's go for it.
So, um, where does bioanalytical fit in the drug development process? This is part 3 of 4. Today, we'll be talking about the overview of PK modeling and the impact on bioanalysis. Our guests are our Vice President of Operations, Lawrence Goodwin, and our Senior Director of LBA, John Pirro. So, why don't we take a moment, and Lawrence, why don't you say hello? And then, John, you can say hello, and then we'll jump right into our agenda items.
Hello, everyone. Thank you for having us on.
Yes, this is John Pirro. Thank you for having us on. I'm very honored to be on this podcast.
So the first topic we're going to jump into, our item, is the overview of PK calculation study design. And really, it doesn't matter if it's small or large molecule, right? And so then the topics we'd like to talk about are compartmentalized and non-compartmental, the Cmax, the AUC, the dosage, and the bioavailability. And Lawrence, do you want to kick us off on your thoughts on the overview of PK calculations?
Sure. I think it's very important that you engage your bioanalytical lab when you're designing a study, especially if it's a definitive TK study. Understanding either if you're going to do compartmental or non-compartmental analysis is very important because they are two different beasts, if you will. With non-compartmental analysis, it's model independent, meaning that you don't have to have a kinetic model, and you're really just looking at in that first order in the plasma, blood, or whole serum. And then if you want to get into compartmental analysis, that's where you're looking at maybe a tissue or a two phase or a two model series where it gets distributed, and the distribution in the tissue is going to be different than it would be in the plasma or the whole blood. And so you really have to have a mathematical model or a kinetic model that will define that movement, if you will. So there's a huge difference. The non-compartmental data will allow you to get your exposure, your systemic exposure, your bioavailability, as well as all the parameters that you listed here, your Cmax, your AUC, and your T half life as well. So it's very important. John, do you want to add anything to that?
Yes, when you're dealing with large molecules, it's very important to understand the ADA or anti-drug antibody effect in your PK analysis because it will affect the PK. So understanding the PK in animals using the ADA assay will help you predict what the new PK could be in your possible clinical trials.
All right, thank you, John. And so stepping back, looking at this, giving some of the definitions, PK really is what does the drug, what happens to the drug when it gets introduced into the body, whether that's through an intravenous route or oral route or an oral gavage or subcutaneous injection. There's different types of parameters. So one of the things when you're setting this up, and one of the reasons why it's important to talk to your bioanalytical team when you're doing this, is you really need to have a finite set of points that you're going to collect, and it needs to be a large enough set that you can actually describe your terminal elimination phase correctly. And that is the phase, the area of the curve at the end of a time course. That's what we do for all the bioanalytical, is a time course of the concentration. So we measure the concentration at various time points. These samples are taken from the animals or from the humans in clinical trials. And then we measure that concentration and plot that and develop a PK profile. And with that PK profile, then you're able to calculate all these PK parameters based upon the model independent nature, and usually use a statistical software package, either WinNonLin, or Watson, or Pharmacokinetica. So those are some of the packages that are out there to do. And so once you have this information, you see this profile, and that profile is going to give you two things. One, if it's an IV profile, you're going to be able to assume that the maximum dose that's going to be in your system at the time in your bloodstream is going to be at time of injection. If you give it another route of administration where it has to then be absorbed and then distributed and then metabolized and excreted, those are the four main principles for PK analysis. You're actually seeing that you will get a rise in concentration, which will lead to your Cmax over time. That's going to be dependent upon how well the compound is absorbed into the system, and that will be considered your Cmax at a certain time point. and then your Tmax will be that highest concentration at that time point. And so that's a very important piece, because then you're able to understand the dosage that's required to get an efficacious dose in the system. And so using that information, you can set up your bioanalytical study to make sure that you have the dynamic range to really cover the Cmax at the upper end and then also at the lower end, so that you're able to define your terminal elimination phase. And what I mean by that is, as you can imagine, a plot, as you go up, it gets higher as time goes up, and then eventually it's going to come back down over time. And then the last 3 or 4 time points after Cmax is really what you need to define that terminal elimination phase. And so you can get the area under the curve, which really is, with Cmax, part of the systemic exposure package that you need for the PK parameters. John, you want to add anything there?
No, thank you. That's fine, Lawrence. I think you've covered everything.
Okay.
Yeah, Lawrence, maybe one quick just for PK dummies, right? So talk a little bit more about Cmax and area under the curve, and maybe you were going to, but just remember, like, if you could just drill into a little bit on how you define the Cmax and what it means and why it's critical, and then the area under the curve for us.
Sure. So as you collect your time points and then they're analyzed, and we actually do the linear regression to calculate what that concentration is at each of those time points, the one that has the highest concentration would be your Cmax. So if you're part of a clinical study and they collect 10 time points from you, ranging from 15 minutes, 30 minutes, 1 hour, and then every couple hours after that, all the way out to 24 hours, the time point that has the highest concentration is going to be considered your maximum concentration. So that's your Cmax. That's how that's defined. And why that's important is because you want to make sure that Cmax is safe and efficacious. That's why they do tox studies in animals, to help you understand what if it's toxic or if it has any potential to cause major side effects or harm individuals. And so the safety is most and foremost. And so those studies are performed to determine that the Cmax is going to be efficacious, but it's not going to be to a point where it can really hurt someone or something. So that's why the Cmax becomes important. And then the area under the curve really is defining that systemic exposure and showing you that it's being eliminated from your system so that you don't have carryover. So I think everybody's familiar with daily dose medication. You'll get, you were talking about azithromycin, where you probably get a five-day pack of pills that you take once a day. Well, if that drug is not eliminated from your system, and then you start taking these pills, you get what's called an accumulation effect. And the accumulation effect could lead to toxicity if you're not careful. And so that's another reason why the AUC is very important. And you have various AUC parameters. You do 0 to last, you can do 0 to 12, you can do 0 to 24 hour. And you can model that using the software and let you know where you're at. And so that comes into play more in TK studies, where you actually have a first day of dosing that you do pharmacokinetics on, where you get those parameters. And then let's say it's a 6-month definitive TK study in rats. And then at 6 months, you do again the same time points. And then you're able to compare and contrast and look at the ratios of what it was on the last day versus the first day. And you'll be able to see if there's any accumulation, and you have to worry about that with your drug. And so all of the drugs that are on the market do this type of testing. It's required by the FDA, and it really sets the stage for safety of our patients out there. And so it's very important. I hope that answered your question, Dominic.
No, it did. That was beautifully said. Thanks. And I think your time, the small molecule timing of this is much different than the large molecule. And John Pirro, maybe do you want to add a little bit around Cmax and AUC and how it's all working for biologics, perhaps?
Yes, that's not a problem. The Cmax with large molecule, as well as the trough concentration of your drug in the plasma of your patients, is very important. It is relying on the NAB assay as well as the anti-drug antibody assays, because it's called drug tolerance. Because we're measuring the antibodies that are naturally formed, polyclonals, in the body, the drug itself, the concentration of the drug at Cmax and trough, is important, because we need to see if the assay can handle the drug concentrations in the plasma, so we can determine how many ADAs, or if there's a neutralizing effect of the drug on the target that you're looking for in the body. So knowledge of the Cmax, trough, and also the area under the curve will help the CRO as well as your PK analysis, because they could be altered, the PK exactly, can be altered because of these NAB assays, NAB antibodies, as well as neutralizing antibodies. So an understanding of how much of your drug at any one point in given time in the body could alter the results from your NAB assay or your anti-drug antibody assay.
Great, thanks.
So, so
Go
another
ahead.
piece that we look at in small molecule is also co-administered drugs and the effect of them on PK as well. Do you want to talk about drug-drug interaction at all?
Sure, we can touch base on that. So there's a lot more combination medicines out there on the market these days. And so one of the things that you do with PK parameters is you're measuring what happens to your drug when it gets into the body as it goes through absorption, distribution, metabolism, excretion. But if you co-administer a drug and you have a cocktail, if you will, um, they behave one way when they're dosed independently, and the testing has to be such that when they're dosed together, you got to see the effect of one drug on the other drug, because sometimes can become an inhibitor or an exciter of the other drug, and that's why some of these cancer medications that are out there are co-dosed, because the one drug actually helps stimulate the other drug to have more efficacy, if you will, and sometimes even more systemic bioavailability and get to the target and be more effective. So we do that testing as well here at KCS. That's something that we always talk to all of our sponsors about, to make sure that if it's a co-administered drug, that they're covering their DDI interaction studies and that those time points are collected. And so one of the ways that we do that, we would help them design their study design where they would dose each independently and have that as a baseline, and then co-dose them, and then use that to see if there's any difference from the PK parameters as a single dose versus the co-dose.
Go ahead, John. You want to add something, Piro?
Absolutely. The industry is now looking towards co-administration of common monoclonal antibody substances with their new antibodies that they're putting out in the industry today. Now, this is causing a situation where anti-drug antibodies could be developed for the first drug that could interact with the new drug that's being co-administered. It's very important that we understand the full PK and Cmax as well as the trough, because these two in large molecule as opposed to small molecule are very important to be able to determine what effect these co-administered antibodies or proteins will have on the PK, Cmax, and also elimination of these drugs in the body. Thank you.
Yeah, no, and John, we just encountered this, right? So I think the small molecule field has been dealing with drug interactions and co-administered drugs for I don't know, 10, 20 years. It just seems like that has been kind of a standard sort of guideline and thing you had to look at. It was definitely something that was covered, but in the large molecule space, it's, I don't know if emerging is the right word, but certainly something that John Piro and I are dealing with much more frequently over the last, say, year or two, in terms of having, as John mentioned, someone might be on a given biologic, for instance, some sort of monoclonal antibody against TNF alpha, and then they're going to be treated with a different monoclonal antibody. That not only does it wreak havoc upon the individual, perhaps, but certainly from a bioanalytical setting, this is something that requires some fair amount of expertise, a fair amount of understanding, and we're blessed with that here at KCAS. So we're kind of wrapping up the main portion here, gentlemen. I think we covered everything. The one thing we might want to touch on is the dosage and bioavailability. Any last thoughts on those items? Maybe Lawrence.
Okay. So sure, with the bioavailability, the way that's calculated through these studies is you will give a control arm, which will be an IV dose, because like I mentioned before, that's going directly into the blood system. And so that's going to be considered the maximum dose is going to be when you inject it. With any other route of administration, especially like oral or subcutaneous injection that doesn't go into the bloodstream, or maybe intramuscular, those have to be absorbed into the bloodstream first. So they have a slower uptake, and then their Cmax will be delayed. Whereas the Cmax for the one injection and right into the bloodstream for an IV intravenous is immediate. And so you're able to take that ratio and determine, if you assume that the IV is going to be 100%, then you're going to be able to measure what it is through an AUC and through the Cmax with the other route of administration. And then it usually is some percentage of that. So the more bioavailable a drug is, means that the faster it gets into the bloodstream. And so you'll hear things at like 50% bioavailable. So it will be at half the maximum. It'll have half the maximum of an IV injection, and the T half life will take longer to eliminate from the system. And so what that does is, most drugs, I hate to say it, from a small molecule perspective, over the last 10 years have been what you call bricks. They're very insoluble. They don't have much bioavailability, and so you have to work at it with formulations and also some changes on the functional moieties around it to increase bioavailability. And fortunately, medicinal chemists have been doing this for a long time, so they're able to add CF3 groups, and they know what other structures to add to really help that. And so they can take a drug that's maybe 2% bioavailable, which means you'll have to dose more of it to get to a systemic exposure concentration that you need to be efficacious, so that the drug will actually do what it's supposed to do, as opposed to if it's an intravenous injection, where it can be a lower dose and have that efficacious level met earlier. But by through formulation development and work of the medicinal chemists working on the structure activity relationship and the analogs that they provide, they're able to move it up to 10 or 20 or 25%. So the more potent the drug is, the less you're going to have to need. And so that's one of the reasons why they look at those assays as well, for potency, and then also secondary assays to make sure that they're only hitting their main target. And so bioavailability is important, because without it, you're not going to have any drug effect once it gets into the blood and gets distributed. And then with the dosage piece of that, it really comes down to the potency of the drug that's going to demand what the potency or the dosage needs to be, so that you get that efficacious level in the system, so that you get that steady state that's going to allow you to have the effect of the drug on your body.
Probably an adjunct to that. A lot of particularly for small molecule, we're doing these studies in phase 1 where it's a PK curve acquired over a short time period. But as we move to phase 3, we're seeing population PK studies where you're taking like one sample maybe every 6 months or 12 months. So how does that figure into the PK interpretation?
So pop PKs are becoming more and more popular, because having somebody in a clinical site, especially in your phase 2 or phase 3, and have them there for 1 or 2 days, depending on what your half life is in the elimination of your drug, to get that whole profile, that's very inconvenient, right? And every day nowadays, everything's fast. So one of the things that's been popular lately is population PK, where you get sparse data sets of a very controlled set and study arm, and you're able to use that data with mathematical formulas and modeling to really predict the effects and get those PK parameters. And so it's just another way of doing it, but it's not, I said it's sparse and it's mathematical modeling. So it's something that's being done, and it takes a real expertise to be able to pull that data out. John, you want to add anything to Pop PK?
Yes, the key thing with all types of large molecule for bioavailability is the amount of time that the drug is occupied and how many times the dosage is being dosed inside the human. The reason for this is bioavailability becomes more difficult to assess, because as you increase the amount of time that the biologic is inside the body, the more anti-drug antibodies are going there. And even though you may have a PK showing that the drug is there at a concentration that you think is bioavailable and also should be efficacious, the actual ability of the drug to do what it's supposed to do may be decreased because of the anti-drug antibodies or the neutralizing effect of the ADAs going forward. So when you're dealing with large molecules, bioavailable has to go hand in hand with the amount of time that the material is in the body, as well as the development of immunogenicity to that drug and how it affects the PK and biodistribution, as well as the bioefficiency of the drug for its target.
Yeah, and well said, John. I think just to round out on the large molecule side, bioavailability is 100%, right? We don't struggle with quite the same sort of as Lawrence very eloquently described, because it's given intravenously, it's taken at 100%. Is that a true statement, John? Do you agree with what I just said there?
Absolutely.
Yeah.
It is one of the paradigms of the large molecules that they are greatly more sustainable in the body, but there's other things that need to be taken into consideration as it goes on in the actual clinical study.
Yeah, absolutely. And so, gentlemen, this has been a fabulous conversation. I'm sure we could sit here for another 30, 50, 2 hours, 3 hours and discuss more of this, but our time has come. Both John and I want to say thank you. You're welcome to have any last words before we wrap up.
No, thank you for having me on and having us on. Really appreciate the chance to talk about this.
And I have to say, I really enjoyed it. So thanks very much.
No, thank you very much for letting me participate, and hopefully maybe in future podcasts again.
Oh,
We've
careful what
got a
you wish
host of
for.
topics.
Yeah. We're always looking for guests, and I'm sure we'd love to talk more about NABs and ADAs, so be on the lookout for that. Um, so, John, let's move on to some feedback, right? Do we have any feedback this week? Oh, we sure do.
We do.
Um,
Shall I start?
yeah, you bet.
Let's go with. So, Gwen says, "Thank you for providing insight on COVID and the bioanalysis side of our industries. I am enjoying the episodes and I missed you last week. Hope you both enjoyed your long holiday break." Thanks very much, Gwen. I have to say, I did enjoy the holiday break. It was first chance to have the family all together in 6 or 7 months, so it was a really nice time off.
Oh, wow, that's great, John. I did get a nice little break. I got to go to visit my in-laws in Nebraska, and that was a lovely time. I always like going there because I don't have to do anything. I get fed. I don't have to worry about much, and it's a nice little respite for me. So that's great to hear that you had gotten together as a family. Yeah.
And
Then
Nebraska's still in one piece despite all your explosives.
Well, I tried. I put on like a nice 2.5, 3 hour show. I mean, it was. I think I mentioned to you, a friend of mine knows somebody that sells the fireworks wholesale. So we got couple like 5 or 6 of these big ones that I never buy, but it was the best show I put on, and I had a lot of fun doing it. The weather was beautiful. It was maybe high 80s, which is about 10 degrees cooler than it normally is. And yeah, all my digits are still intact. No one got hurt. There was always a periodic little bottle rocket kind of getting a little too close here and there that scares my mother-in-law, but overall it was a real lot of fun. Yeah, it was a blast, literally and figuratively.
Excellent.
The next feedback we got is from Brian. Brian says, "I'm with John. I can't stand the ongoing gray noise about hydroxychloroquine. Can we just all agree once and for all that if you'd like to take it, take it. Otherwise, just stop talking about it." Well, that one kind of makes me laugh, right? So, because now, John, we don't. We thought we had put a nail in the coffin with the Lancet study, but apparently, I don't know. I saw some grumblings that people are still saying it's quite effective, but let's shelve that for now, or if you want
I,
to add some
I, I
thoughts on it.
I propose ignoring it until we see someone actually show data that isn't criticised by the peers. We haven't seen anything yet to that effect. And I have to say though, I wouldn't say if you want to take it, take it. I mean, to me, it's like drugs are there to help you. If you've got a condition, I don't really like introducing these alien things into your body if they're not there for a reason. If it's not having any effect, don't add it. I'm yeah, that's the way I view most
Yeah,
medicines.
we agree. Last one, wrap us up, John.
Yeah. Last but not least, from Dion, I'm enjoying listening to both of you and podcasts. Thank you. Thank you very much for your feedback, Dion, and anyone else who wants to give us feedback. We're always happy to discuss anything you might want to talk about.
Yeah. So, John, it's as we're wrapping up here, it's a thirsty Thursday. Any last things going on in your world? Or I know I still can't find yeast, although I think I told you I did get some. So this week, pizza time is back. That's a shout out to our BD rep, Jeremy Rochford, who likes to call it pizza time because he knows I love to make the pizza. But yeah, I'll be making some pizza this weekend. What about you?
Today is my daughter's birthday, so we'll be doing something to celebrate that. So I'd actually like to spend some time away from my desk today if I can. Yeah, I sort of did a couple of interesting recipes over the weekend. One with, because my wife is on one of the where she gets the regular shipment of vegetables, and we got kohlrabi, which I've never ever cooked with before. Found a recipe which was basically cream, milk, thyme, thinly sliced kohlrabi, top it with cheese, stick it in the oven, which sounds great and easy, and it was, and it tasted delicious. However, in slicing the kohlrabi thinly, I found I realised how much mandolins don't like me, and I am now nursing a widely cut open thumb. But yeah, I shall be taking it easy on the culinary front this week.
What were you cutting again? I don't know if I know what that is.
Kohlrabi. It's I don't. It's a root vegetable, so it's spelled Kohlrabi, and it's sort of quite a tender thing. It sort of sweetens up with cooking, and yeah, I've never done it before. We've sort of had it with these baskets before, and it sort of sits until we never do anything with it, and then we sort of throw it out. This time, I made sure that I actually cooked with it. So, actually, it was
Yeah,
from a recipe book by my favourite chef in the UK, a guy called Simon Rogan. So, yeah, I can't get to
you're
restaurants
way
to cook his recipes.
You're way more sophisticated than me, John. I made some nice homemade mac and cheese this weekend. That was delicious. I don't know. Yeah, and then now I know why I've never heard of it. It's a vegetable, which mine includes. I like the vegetables, but they're pretty basic. I make a good. I love bacon, Brussels sprouts. That's one of my specialties. I use a little rub, barbecue rub with it, and that's a good one. But that's great stuff. Now I've made myself hungry. But and that's great. You got a birthday coming up. I have to deal with a tree that's dying outside. I got a beetle issue that I'm going to
Oh
have to
wow!
deal with this weekend. And I'm also looking at trying to replace my deck. So I don't know if you've ever done anything like that, John, but what a headache trying to get new parts for the number of boards and railings. And oh my God, it's exhausting. I just wish it was like, give me two, let me pick one, but it's
No,
quite
we don't have anything like that, but we have an old house. I think it was 1870 it was built, and I closed the front door yesterday, and it's like an external door with a loose glass window in it that fell out onto our porch. So that now needs replacement. So nothing
okay.
major on that. And my beetle problem is Japanese beetles on my strawberries. But anyway, let's move on, wrap
Yeah.
up, and give a cue for next week.
Yeah, so we're going to be wrapping up our series on where does bioanalytical fit in the drug development process. We'll be doing part 4 of 4, and that's going to deal with how much bioanalytical is needed for the biology and mechanism of action. We're going to touch on discovery. We're going to touch on biomarkers and the PD frenzy, and then we'll look at ADA and safety. That's a wrap for me, John. Any last words before we call it good for week number 13?
No, I'm very glad to wrap this one up, and thanks again to our guests, Lawrence Goodwin and John Piro. Look forward to doing this next week.